Hi everyone, my name is Dr. Huang, and I'm super excited to share this brand new lab from miniPCR Bio with you all, a classroom-friendly lab that visually teaches both protein expression and purification. My friendly moderators will be in the chat answering questions and providing any relevant links, so feel free to ask any questions you have there. Let's just jump right in. If you're not already familiar with us, our mission at miniPCR Bio is to make science accessible for everyone everywhere, whether that's out in the field, in the Amazon jungles, in the forest foraging for mushrooms, or in classrooms like yours. And the team that creates the labs and curriculum for your classrooms are actually made up of both scientists and former teachers who are all wonderfully nerdy, so we can deliver a high quality curriculum that actually fits your biology teaching needs. So you may be familiar with our hands-on learning labs where biology concepts are emphasized while using biotech as a tool for demonstrating those concepts and illustrating how they can be used to solve real-world problems. Today, we're going to be introducing a new type of learning lab, biotech skills labs. The main focus of this biotech skill lab is the biotech tools and techniques and methods themselves. The goal with these labs is for students to envision their future in the biotechnology industry. Students will gain hands-on experience with essential biotech methods, hone skills like laboratory math, and explore new career paths. So in this webinar, I'm going to be walking you through our new protein expression and purification lab. Those are two of the most critical and crucial techniques in biotech, and I will show you how you can use this lab to help your students understand how they work. The goal of this lab is to use protein expression to make a protein you want, then use protein purification to separate that protein of interest away from everything else. It's very classroom friendly. It uses proteins that can help your students track the entire process visually. Plus, it just takes 2 class periods with very minimal equipment needs. So after this webinar, I hope you will be able to understand why protein expression and purification are essential in the biotech industry and how to teach those techniques in the classroom with this new lab that I will demo today. So why do I keep saying that these two techniques are important in the biotech field? Well, from biology, we know that proteins are extremely useful and can perform a wide variety of functions. So being able to make proteins such as insulin to help treat diabetes patients is important. In biotech, scientists use the term protein expression to describe the process of making useful proteins. Now, simply making the proteins is not enough. If we're making insulin, for example, we need to make sure that the protein doesn't contain impurities, since we're going to be giving it to patients. To separate the protein away from everything else in the expression process is called protein purification. To learn more about why making and purifying these proteins are important, I did write a blog post on proteins that change the world. If you want to see more examples, we'll link it in the chat down below. So typically in industry, protein expression and purification are done at a huge scale. You can see from these images here, imagine giant equipment that are hundreds, even thousands of gallons large, where you can't see what's going on. Obviously, you're not going to be doing that in the classroom. So what we've done with this new lab is to scale down the techniques to a point where they are affordable and easy enough for your classroom, but also designed to emphasize the principles of what your students need to know about protein expression and purification. So whether they encounter these industrial versions in their jobs, they'll be able to apply what they learned. So now I'm going to be walking through our lab while explaining the techniques behind it. Let's start with the first part of the lab, which is protein expression. To make useful proteins, like the insulin example we keep bringing up, Scientists typically use living cells to produce almost any protein they want. They do so by introducing DNA that encodes the protein that they want to make to the cells, and then the cells become these little factories that make lots of that protein. To make this lab more classroom friendly, instead of live cells, students will be using BioBits, which is our cell-free protein expression system. Briefly, BioBits contain all of the molecular machinery, things like the ribosomes, the amino acids, all that fun stuff needed for protein expression. If you want to learn more about BioBits and cell-free systems, this is actually our fourth learning lab that uses BioBits. We do have other ones that cover the central dogma, protein structure and function, and antibiotic resistance. If you want to check those out, we'll also link some blog posts and readings down in the chat that explain BioBits in more details if you're curious to learn more. But for today's webinar, all you need to know is that BioBits come as these freeze-dried pellets in tubes, and all students need to do is simply add the DNA that encodes the protein to rehydrate the pellets, and your protein of interest will be made. So I'm going to switch to camera mode now to show you how easy that is. All right, so first let's get my gloves on because it's always good to practice good PPE. So give me a second to pop these on. And I'm just going to be showing how easy it is to rehydrate the BioBits pellets using DNA. All right, so that's one on. Let's get the other one on. And I'm going to be holding up to the camera now these BioBits pellets. So you can see here, I'm going to hold my hand up. You can see here these yellow pellets in the tube. Those are the BioBits pellets that will make our protein. And all I'm going to do is to add DNA to rehydrate these. So I'm going to grab my pipette. I'm going to grab some DNA. All right, so I got DNA in my pipette tip. And I'm simply going to add those to my pellets. So again, holding this up, you can see that I'm simply just going to stick my pipette tip in. I'm going to let it rest against the inside of the wall, and I'm going to push out my liquid, and you can see there, all I did was just add the liquid to that first tube, and you can see the pellet is rehydrated. So in case you missed that, I'm going to do it for the other three as well. So I'm going to the second tube here now. I'm going to hold it up so you can see it. Pipette tip in, just add your DNA. It drips down and rehydrates the pellets. Two more times. DNA, pipette tip goes in, rehydrate the pellets. All right. And I'm just doing this for demo purposes, but just, make sure you switch up pipette tips between reactions. All right, so as you can see here, if I hold my hand up again, those yellow pellets are all rehydrated into solution. All right, so all I did was add DNA to rehydrate these pellets, and all your students have to do is just leave this overnight at room temperature, and when we come back the next day, all the proteins will have been made. Isn't that awesome? So easy to do. All right, let's get back to the slides. All right, so the protein of interest in this lab will be green fluorescent protein, or GFP for short. Your students can think of GFP as the insulin that the scientists want to make. And the reason why we are expressing a fluorescent protein is to make the process visual for your students and allow them to track where the GFP is at all times. Now, GFP is not going to be the only thing in the tube after it's expressed. You can ask your students, what else is going to be in that tube? I'll pause and I'll let you think. What else is going to be in that tube? Now, I'll give you the answers now. There's going to be all that cellular machinery from the BioBits, right? Like the ribosomes and the polymerases, as well as that DNA template that we added. So all this other stuff is clear and or too small to see. So how can we track where all this stuff ends up? To do so, we're actually going to also make a red fluorescent protein, or RFP for short. And that's going to be a visual indicator of everything else you don't want. Side note here, the images you see in these slides and in our lab guide are actually optimized for color vision deficiency, which is why the RFP protein here looks pink in the image, even though they're red in real life. All right, so both RFP and GFP will be expressed in the same reaction, and when green and red fluorescents are combined, it actually appears yellow. So when you see yellow fluorescence, you know your GFP, your protein of interest, your insulin, is contaminated with RFP and everything else that you don't want. The goal of the lab on the purification side is then to learn how to separate the GFP away from everything else. Okay? Hopefully you're all following me so far. Let's take a look at what the reactions look like. Your students will actually set up 4 different protein expression reactions as shown here. The first 3 tubes are going to be reference tubes. Tube 1 shows you what GFP only looks like. Tube 2 shows you what RFP only looks like. And tube 3, what the combination of green and red fluorescence looks like. Tube 4 is a duplicate of tube 3 as it has both GFP and RFP, except that's going to be the tube we actually purify and separate the GFP away from everything else. Okay, I'm going to switch back to my camera so I can show you what your samples will look like in real life after you've added the DNA and let the expression go overnight to room temperature. So I'm going to pop them in my viewer here. All right, so here's tubes 1, 2, and 3. With my okay, here we go. Tubes 1 with the GFP, tubes 2 with the RFP, and tube 3 with the combination of GFP and RFP. Again, those 3 tubes are going to be your reference. And tube 4 on the end here is what I'm going to purify live on camera for you. Again, it's just a duplicate of tube 3 as it has both GFP and RFP. Okay? You might notice that I'm viewing the fluorescence in this box here. This is our P51 viewer. It's got blue lights that shine up at the tubes. You can see here. So you'll be able to visualize anything that has fluorescence in these tubes through the front window right here. We do have other labs that use the P51 and resources that explain how fluorescence works. So we can also link those in the chat if you want to learn more about it. All right, let's get back to our slides to explain a few more things. So now that we have made the GFP, our protein of interest, our insulin, we need to isolate it from everything else in the tube. This is a process known as protein purification. To do so, we can use unique characteristics of the protein, such as their size, their charge, their hydrophobicity, to pick out things we want away from the things that we don't want. In this lab, we're actually going to be using the most specific purification technique to separate our GFP away. The GFP used in this lab was actually engineered to include a tag that's made up of a few amino acids. We're also using these tiny microbeads that are coated with a ligand. This tag fused to the GFP binds very specifically to the ligands coated on the microbeads. So these beads, using the ligand, can grab onto the tag, which is attached to the GFP, so therefore the GFP is now captured and bound to these microbeads. This method is actually called affinity purification, and we're using it for this lab because it's the most specific method, because nothing else in that BioBits reaction has that tag, which allows your students to get very pure results easily and quickly. So once the GFP is bound to the beads, we need to hold the beads in place while removing everything else. Well, these beads happen to be magnetic. Again, another easy to use method to give your students a good protein yield at the end. We'll simply hold the magnetic beads in place with a magnet and then use the pipette to suck up everything else. I'm going to show you how easy this is on camera again. Pull up that. All right. To show you in person how the magnetic beads work, here are the beads in buffer. There's no proteins yet. It's just the magnetic microbeads in buffer. All right, so you can kind of see that my solution, there's a little pellet at the bottom. That is where the magnet beads are. All right, so now I'm going to stick it in this tube rack that has magnets embedded inside of it. This is our new Pull-It magnetic rack. So when I stick my tube in there, I'm going to tilt it to the side so you can see. Hold up to the camera. All right, so you can see that the beads are the beads are just magnetic and they're sticking to the side of the tube close to the magnetic rack, right? You can see that the beads are at the side closer to the rack right here. Move that again. Yep, you can see that. Which means I can just reach in with my pipette tip and suck up all the buffer. So let me grab a pipette tip, set my pipette. If you hear meowing in the background, my cat has joined me for the hands-on portion. She's sitting on my lap right now because she also loves science. All right, so you can see the magnetic rack right there. And I'm just going to stick my pipette tip in, suck all the buffer out. You can see all the buffer is being sucked out, but the magnetic beads are left behind because you can see it's dark there. All right. And my cat has definitely joined me on my lab bench. Hello, kitty. So this Pull-It is easy to use, is designed specifically to make it easy to pipette in and out of, and is much cheaper than other magnetic racks you might see on the market. We're going to put more info in the chat if you want to learn more. And my cat Pancakes loves the new Pull-It, don't you, kitty? All right. Why don't you get down? Thank you. All right, back to the slides. So protein purification can be broken down to 3 main steps. Virtually all protein purification processes, no matter what protein you're purifying, no matter what technique you're using, will all use these 3 steps. So it's important for your students to understand how they work. So I'm going to explain each step in detail and show you how on camera how it will work in the lab. So let's just jump right in and start with step 1, which is binding. Binding is where we're going to actually capture our protein of interest, again, the GFP, and hold it in place while we remove everything else represented by the red. So here's where the tag on the GFP and the magnetic beads will come into play and bind together. So as a recap, your students will add their protein expression mixture to the magnetic beads, let it incubate, and then place the tubes in the magnetic rack. It might be good to emphasize to your students at this point that it's only the beads that are magnetic. Students might make the misconception that the GFP itself is magnetic. It's not. The magnet is only to hold the magnetic beads in place. The GFP just happens to be stuck to the beads via that tag ligand interaction. Everything else, because it doesn't have a tag, will still be free floating in solution. All right, so I'm going to switch back to my camera and I'm going to show you a sample that I have that I already mixed together the night before. This is already have my beads and my GFP RFP reaction mixed together and incubated for a bit. You should let it sit for at least 30 minutes. And if you leave it overnight, like I did, you can get even better results, a better yield. So I'm going to put this in my pullet rack as such, okay, to capture the magnetic beads. And before I use my pipette to pull off all the stuff that we don't want free floating in solution, I'm going to show you an optional but really cool step to visually see the GFP separated from the red in the same tube. All right, so I'm going to hold up my P51 and try to get that on screen there. All right, so I'm going to grab my magnetic beads. And if I transfer this to P51 and get the camera focusing, you can see the GFP. Where is my P51? Okay. You can see the GFP stuck to the magnetic pellet at the bottom and the red still in solution. Don't know if you can quite make it out, but I'm going to do that again. It's kind of hard to see on camera, but in real life, it's really, really cool. All right. Okay, sticking that in again. And you can see in that tube there, you can see red, and you see that green splotch in the middle right there? That is the GFP stuck onto the magnetic pellet. So, since it didn't come through on camera that well, I'm just going to quickly show it. Here it is what I was showing. You can see in the same tube, you can see the magnetic pellet on the bottom, which appears green because of the GFP bound to it, as well as everything else floating in solution represented by the red. All right, so that was what you should see in real life. It is really cool to be able to see the green and the red separated in the same tube at the same time. That's my favorite part of the lab, actually. All right, so I'm going to take those tubes and put it back into my pullet here. And I'm going to use my pipette here to remove the red, okay? I'm gonna set my pipette. All right, so now I'm actually gonna go back in the pullet and remove the red and everything else we don't want, while the GFP, the insulin that we actually want, stays behind. Okay, so I'm just gonna tilt this to the camera so you can see. Ooh, there we go. I'm gonna stick my pipette tip in. Just gonna hold it up and I'm gonna suck up all the liquid in there. All right, so I removed all the stuff we don't want. I'm actually gonna keep the stuff that I pull off. This is called a fraction, and it's important to save each fraction just in case something goes wrong. All right, so I'm just gonna take that fraction and I'm just gonna plop it in the empty tube next door. And all these other tubes are gonna take other fractions throughout the lab. All right, so if I actually stick this into the P51, I didn't get all my liquid. Let me just quickly fix that. That's what happens when I try to pipette and talk to the camera at the same time. There we go. So if I put this into the P51, we can see that that fraction that we just pulled off in tube number 2, it's red. So I think it's always a good idea to stop after each step and ask your students to check their understanding. For example, at this point, it would be a good idea to ask them before you show them in the P51 what color fluorescence that fraction we just pulled off will be. Right? Because only the GFP has the tag that binds, everything else in the reaction, including that RFP, will be removed, so the fraction will appear red, which is what you saw on camera in the P51. So let's actually compare that fraction back to our reference samples. Again, we have tubes representing what green only, red only, and the combination of the two look like. Ask your students, how does the fraction compare to these references? And you can also ask them, what does this mean for our purification process so far? For example, here we can see that our fraction looks mostly like the RFP only tube, which tells us that it's mostly RFP in that fraction and that most of the GFP is bound to the beads. That means we have removed most of the RFP and the other unwanted stuff while leaving the GFP behind. On the other hand, hypothetically, if this fraction looked more yellow than red, that would mean that there is still a good amount of GFP that didn't bind to the beads, and perhaps we would need to either redo this step or do it for longer. All right, so now let's move to step 2, which is the wash step. So after you remove the unbound fraction in the last step, there is likely still some stuff we don't want, just some residual things stuck to the sides of the tubes and such. That's why we have a wash step to remove all of that. To do so, we'll simply add a wash buffer to the reaction as a way to rinse off the leftover RFP and anything else that we don't want that maybe was left behind. All right, so switching back to the camera, I'm going to take that reaction and I am going to add some wash buffer. All right, so pulling up some wash buffer in my pipette tip here. And I'm adding the wash buffer to my tubes. And you can see I'm not putting my tubes in the pullet right now because I want it to mix around in there. All right. As you can see, the motions here are basically identical to the previous step. We're adding some stuff into a tube, then we're going to take such tubes, we're going to put them onto the pullet, and then again, we're going to use our pipette tip to pull off the fraction, and this time this is the wash fraction. So I'm just going to stick my pipette tip in and whoop, yank everything up. All right, and again, I'm going to keep this fraction. We always want to keep each one of our fractions. And I'm going to just put it in the tube next door. Make sure I didn't get all of it. There we go. All right. So now we're going to take a look at what that fraction looks like. I'm going to put it in the P51. And when we do so and hold up to the camera, you can see the red tube from our last step, and you can see the tube next to it has liquid in it, but it's clear. Again, since the motions are really similar in terms of these steps, it's again a good idea to quiz your students, pause, and check their understanding to make sure they understand what's going on. Here, ask them again what color fluorescence that fraction we just pulled off in the wash will be. Right, because we remove most of the stuff that we don't want in the previous step, including most of the red, that fraction should look mostly clear or just very faintly red. Let's compare that fraction to our reference samples. Here, since our fraction is clear or only faintly red, this tells us that between the bind and wash steps, we have removed virtually all of the stuff we don't want, which we are tracking with the red fluorescence. Note this step, if your students didn't manage to pipette off all the liquid in the previous binding step, the wash step is a good place for them to help them to get the little last bit that they might have missed. All right, so let's now move on to the last step, which is elution. So now that we are sure that there is barely any RFP or any other stuff left, we want to claim our GFP and get it off those beads because we're not going to be, injecting the insulin stuck to the beads into a patient. We're going to be doing so by adding an elution buffer to the reaction, which gets the GFP off the beads and into solution. Another little pop quiz here. What do you think is in the elution buffer that allows for this to happen? The answer is that there's actually several ways that this can happen, but in this specific lab, the buffer has a competitive binding element for the ligand on the beads. What that means is that this molecule that I'm showing here in orange will bind to the ligand on the beads and displace the GFP into solution and allowing us to collect it. All right, so I'm going to show you this last step on camera. We're going to grab the tubes that we've been working with, and I am going to add an elution buffer to that last tube. All right. Make sure my camera is okay. All right, so adding an elution buffer to that last tube. Again, the motions are similar to what is happening, but what's happening molecularly is different. All right, so I've added an elution buffer to the magnetic beads. I'm actually going to let this sit here for a few minutes while we do one final question together, giving the elution buffer some time to work. And that question is, again, to ask what color fluorescence that fraction we just pulled off will be in the elution step, right? Because we added the elution buffer that will displace the GFP into solution and allow us to collect it, the elution fraction will appear green. Again, let's compare that fraction to our reference samples. Here, since our fraction is green, this tells us we have successfully collected our protein of interest, the GFP. And since it only appears green and not red or yellow, that indicates that we have successfully purified it away from the red and everything else that we don't want. All right, so let's take a look at that. So I got my samples here. So completing my reaction here, I'm going to put it in my pullet to hold the magnetic beads in place and pull off the elution fraction, which should have our GFP in it. I'm going to move that fraction over to the last tube. Again, we're keeping all of our fractions. And again, you can see all these motions are similar, putting the tubes in the magnetic rack, holding the beads in place while you pull off the solution. All right, so let's take a look at it in our P51. And we can see, I'm going to line it up. Yeah, we can see that last tube is green. All right, I know it looks really faint on camera, but in real life, it's much greener than what you see. Yeah, you can definitely see that. And here are the pictures of what we expect. There we go. So we can talk through it. You can see here that we have separated everything we don't want, shown in the red and the unbound and wash fractions, from the GFP that we do want, shown in the green in the elution step. Okay, so you might be asking, hey, that green doesn't look as bright as the reference samples. It didn't look that green on camera, and in the picture it doesn't look as green. And that's actually completely normal. You're never going to be able to purify 100% of your protein of interest that you have expressed. The amount that you're able to purify is referred to as yield. And in industry, a yield of 50% is actually considered pretty decent. This is because we often sacrifice the yield to make sure the protein is really pure. If we go back to the insulin example, we would rather give patients a very pure and safe insulin than try to maximize our yield and end up with a less pure sample. So we do actually have more discussion questions in our lab guide that goes into this, including how to calculate purity and yield of a protein expression and purification reaction. In terms of time scale, this lab only takes 2 class periods. The first class period is to set up the expression reactions and let them go overnight. The second class period is to actually do the purification process, which you saw me do live here today. And this is amazing because similar classroom activities that cover expression and purification can take more than a week of your class time. A few notes here on the timeline. First, the class periods do not need to be consecutive. Once the proteins are expressed, they are stable for a few days if you don't get to the purification the very next day. Second, if you want, you can actually break this lab into 3 class periods by letting the GFP bind to the beads overnight instead of the minimum of 30 minutes, and this actually helps improve the yield. So it just depends on your class time and what you're looking for. All right, and as you saw from my demo today, you don't need that much equipment to complete this lab. You'll need P20 and P200 pipettes, which we do have for $59 each. You'll need a magnetic rack. which our pullet is only $38, and you saw on camera, it's super easy to use. And you'll need a blue light viewer to see the fluorescence, which our P51 makes this super easy, and they're only at $35 each. As for the lab reagents themselves, they are $119 for 8 lab group. So if you have put students in groups of 4, that is up to 32 students a kit. And it comes with all the reagents your students need to express the proteins using our cell-free BioBit system, and also to purify the proteins using magnetic beads. If you don't know, all of our curriculum for our hands-on labs are free. Just go to the product page, which we will link in the chat down below so you can access it now. Select curriculum downloads as shown on the screen here, and you can access the teacher guide, the student guide, both in PDF and Google Doc form, and classroom slides that are similar to these slides that I showed today in PowerPoint format so you can customize them for yourself. That way you can review the lab, go through it, look through the protocol before you buy. And again, in case you forgot everything that I went over today, you can read it all in the lab guide. The curriculum also comes with career profiles of real-life scientists working with proteins using these techniques of protein expression and purification, their unique career paths, and the projects they work on now to inspire students in case they are thinking about a career in biotech. Plus, lots of discussion questions that allow students to practice lab math and how to calculate things like yield and purity. And that's it. Hopefully you guys had a fun time seeing how our new protein expression and purification lab worked, how easy it is to do these techniques at a small classroom scale, and how students can better understand the process by tracking them visually. Thanks for joining us today, and I can't wait to hear how this lab goes in your classroom. Thanks. Bye.